great pleasure to be here. We have a very exciting session for you today. Science magazine described cancer immunotherapy as the breakthrough of the year, last year I think it was. And so this is the only session on cancer immunotherapy that you're going to have at this meeting. So for those of you who are not familiar with the field, I'm going to give you a little very brief introduction, then we'll go into our talks. And there's a slight change in the program, of the order of the program. Dr. Beavis is going to speak before Dr. Darcy. I'm hoping, and particularly since we have a relatively small audience, that we can have some questions and some discussion at this session. And so I'm not going to take questions after my talk because I'm going to hand it over to my colleague, Nicola Mason. My talk is very much an introduction to Dr. Mason's work. But then hopefully we'll have time for some questions after those two talks. We'll try and take some questions after Dr. Keeper-Evans' talk, Dr. Darcy's talk, and then after Darcy and Dr. Beavis' talk. We'll run from Dr. Beavis into Dr. Darcy's talk, take some questions there, and then also at the end for Stefan Eichmuller, if we can all keep our presentations down to a reasonable level so that we have time for questions. I think that would be most informative. So I just got a couple of slides to explain to you what cancer immunotherapy is, in case you don't know, because I know this is a very broad meeting, and I've been talking to like plastic surgeons, all kinds of folks who work in areas that I know nothing about. And there are basically two flavors to cancer immunotherapy. One is adoptive immunotherapy, and this is where you expand the immune system cells outside the body and then transfer them passively back into the host for therapeutic purposes. And Dr. Darcy and Dr. Beavis are going to talk about their work using the chimeric antigen receptor T cells, or CAR T cells, which have become really a huge amount of news about. That therapy is not yet in the clinic, but it's getting close. We're on the cusp, I hope. By the clinic, I don't mean clinical trial, I mean accepted by the appropriate regulatory bodies for use routinely. And the other flavor of adaptive immunotherapy is passive transfer of engineered antibodies, which nobody's going to talk about today. But in fact, most of the currently approved immunotherapies belong in that category, passive, and I'll show you a few examples. Active immunotherapy is when you're actually using the host's immune response itself, and you're trying to jumpstart it so it recognizes and kills the tumor. That's the field I work in. It's sometimes called cancer vaccines. Dr. Mason is also going to tell you about her work in that field using the technology that I developed, the Listeria technology. And Dr. Keeper-Evans also works in that field, and he has a very unique spin on active immunotherapy in that he makes synthetic antigens for which will hopefully provide a better immune response than the natural antigens that a lot of us target. And you're going to hear a lot about HER2/neu here because many of us are targeting HER2/neu for both adaptive and for active immunotherapy. And then a really important part of cancer immunotherapy is you can't do cancer immunotherapy, you can't prime the immune system against cancer if you don't know what the antigens are that tumors express. And so a very important part of cancer immunotherapy is antigen discovery. And Dr. Eichmuller and Dr. Keeper-Evans both work in that field, and their talks will focus somewhat on that. And antigen discovery serves both adoptive and active immunotherapy. Adoptive immunotherapy because it can identify cell surface antigens that CAR T cells can be engineered towards and recognize, or it can provide antigens for cancer vaccines. And so what is a cancer vaccine? So a cancer vaccine is really just a way to harness the immune system to tumors by active immunization, using very often the similar kind of approaches that we use for vaccines, prophylactic vaccines against infectious diseases, the shots that we all get as children. However, most cancer vaccines are applied therapeutically. There's actually only one or two cancer vaccines at the moment that are prophylactic, and they target, obviously, cervical cancer, human papillomavirus as a prophylactic vaccine for, and hepatitis, so the hepatitis vaccine for that. But all other vaccines so far are being developed therapeutically. But we think that as we learn more about the genes that predispose individuals to cancer vaccines, eventually it may be possible to make prophylactic cancer vaccines. So this is not a complete list. I did my best with this. But these are the FDA-approved accepted forms of immunotherapy for cancer at the moment. And I've color-coded them. So the ones in pale blue are the so-called checkpoint inhibitors. These are antibodies that block immune system molecules that tend to damp down the immune response. So by blocking the activity of those molecules, that allows the immune system to develop. Some of them are targeting cell surface receptors. You know, famous one is rituximab, which is anti-CD20 that binds B-cell lymphomas and then by antibody-dependent cytotoxicity kills them. So they're all in green. Then there's the therapies that target growth factor receptors, such as Erbitux and Herceptin, which are in orange. And then there's some older therapies that have multiple different effects that are in black. But what I want to point out is, where is this pointer? We have a pointer, right? This is a pointer. Good. Oops. I have to point in the right direction. That's the only problem. Here we go. What I want to point out is this here, Proven. This is currently the only approved cancer vaccine that we, if I got that right, then he's going to speak up and say, no, it isn't. No, I'm absolutely confident that this is the only approved cancer vaccine, and it's a pulsed dendritic cell vaccine against prostate cancer. It's approved. I'm talking about the United States. I'm not talking about countries other than the United States. So I think there are going to be other cancer vaccines that are going to be approved shortly, and I'm rather hoping that mine is one of them, of course. And there's a lot in clinical trials and in phase 3 clinical trials, but that currently is the only approved one. So I'm going to move into my own talk, which is on using HER2/neu as a target for Listeria-based cancer immunotherapy. And I'm going to focus on breast cancer for my talk. And so what is Listeria? This is a bacterial vector. So the idea here is that if you should be able to jumpstart the immune system by making tumor antigens, which tend to be tolerogenic, recognized by the immune system by wrapping them up so they look like a pathogen. So you put them into a pathogen. And the pathogen that I use is Listeria, and I think I have really quite good reasons for that. I've been working on this for over 20 years, so I should have good reasons for it. And one is that it has some rather unusual properties. So this is a gram-positive intracellular bacteria, so facultative bacteria, so it can live outside the cell. You can grow it up in nutrient broth in vats. Very easy to manipulate. Not so easy to manipulate genetically, but you know we've developed techniques to do that. It's a foodborne pathogen. Many of you have heard of listeriosis. But the interesting thing is it has this unique life cycle, which is ideal for the presentation of antigens. So Listeria gets taken up by phagocytic cells, and many of these are antigen-presenting cells, dendritic cells, macrophages, and they get taken up into a phagolysosome. And most of the bacteria are actually killed in that compartment, where the antigens that are released can egress to the surface of the cell to activate CD4 T cells. But a proportion of Listeria has virulence factors, and particularly one called Listeria lysine O, or LLO, that allow it to break out of that vacuole and live in the cytosol of the cell. It does two things when it's in the cytosol of the cell. One is that it can actually reorganize host cell actin and spread from cell to cell. I realize you can't see this micrograph at the back of the room, but what it's showing you is Listeria living inside a cell and actually spreading from one cell to another. Here you see by way of membrane fusion of the two cells. So using actate it spreads from cell to cell. Any antigen that Listeria produces and secretes into the cytosol can target the class 1 pathway for antigen presentation, egress to the surface of the cell, where it can activate CD8 T cells. So what you get in terms of the immune response to Listeria is a very strong cell-mediated immunity, hardly any humoral immunity, because once it gets in a cell, it never has to get out again. In addition, you get a huge amount of innate immunity to Listeria, and I'll talk a little bit about that later. So in terms of, obviously you can't use Listeria, wild-type Listeria in a human being. It is a pathogen. So we genetically modify it, and we attenuate it. And we delete the ACT-A gene that allows it to cell-to-cell spread, so it can't do that. We also delete genes that are required for D-alanine synthesis, and D-alanine is required to make bacterial cell walls. So Listeria actually cannot live without the genes that are required for D-alanine synthesis. And so we also have genetically modified it to express the antigen of interest, and we use a plasmid-based strategy. And the antigen is fused to Listeria lysine O, which is a Listeria virulence factor, and that acts as a pump, so it acts as an adjuvant to the antigen. And the plasmid is maintained both in vivo and in vitro because we put a copy of the DAL gene on the plasmid. If the plasmid is lost by the bug, it can no longer grow. So this enforces retention of the plasmid. There is no antibiotic selection marker, so this makes this very safe for clinical use. We do not need it for the transformation strategy because we can select for transformation by the ability of the bacterium to synthesize cell walls in the absence of D-alanine. So we find that this very attenuated strain is cleared very rapidly, and is actually cleared both in wild-type mice and interferon gamma knockout mice by innate immunity. So this is a wild-type mouse, and this is the gamma knockout mouse. And we also, I don't have it on here, but we've also looked for its clearance in skid mice. So it's a very safe vector, and it has now gone into humans. We have a number of human clinical trials, which if I have time, I'll tell you about. So, you know, the reason why these vaccines are so effective is that you get a very strong burst of innate immunity early in the immune response, and this jump-starts the immune response particularly well to tolerogenic antigens like tumor-associated antigens, many of which have homology with self-proteins, and therefore there's been central tolerance and peripheral tolerance to them. And so you get a Th1 pattern of cytokine and chemokine release, you get upregulation of co-stimulatory molecules on the surface of the cells that have been infected, DCs and macrophages, and you get a burst of cytokines released from infected myeloid cells very early in infection. And these stimulate NK cells to make interferon gamma and IL-12, and this promotes the maturation of dendritic cells and skews the response to a Th1 response. I've already told you the kind of adaptive immunity you get is a very strong CDL response, even in the face of tolerance. And I can show you some data of that in mice. It tends to stimulate tumor cells. Interferon gamma that's produced can stimulate tumor cells to produce chemokines that recruit chemokine receptor expressing CTLs in the tumor. So you always see a huge infusion of CTLs into the tumor after vaccination with a Listeria construct. We also find that it recreates the tumor microenvironment, minimizing Treg cells and inhibitory cytokines, and reduces the number of myeloid-derived suppressor cells in the tumor as well. So but not in the periphery, interestingly enough. So it really sort of primes the tumor microenvironment to be acceptive of the CTL response that then goes into the tumor and kills the cells. So I'm going to skip over this. So let's talk about what we've done with HER2/neu and how we're using this for the immunotherapy of breast cancer and osteosarcoma. So HER2/neu, I think most of you know what HER2/neu is. It's a large transmembrane epidermal growth factor receptor protein. It is expressed, it's a self-protein, it's expressed in skin, mammary glands, cardiac tissue, neuronal tissue. But it accumulates mutations that allow it to form heterodimers, and these activate signaling pathways and stimulate the tumor to grow. So it promotes cell proliferation, survival, and angiogenesis. So if your breast cancer or any cancer is actually is overexpressed in a large variety of cancers, carcinomas, breast, ovarian, pancreatic, etc. If your cancer does express HER2/neu, then that's not good news because it correlates with an aggressive phenotype, increased metastatic risk. The work that I'm going to tell, so I developed this vaccine a long time ago now, actually for human breast cancer. So this human HER2/neu that expresses, being such a large protein, it has a big extracellular domain, transmembrane domain, and a big signaling domain, and that's too big for a bacterium to make. You know, it's toxic to the bacterium. Bacterial proteins tend to be small, and it's hard to get them to make anything much more than 500, 600 residues long. So what we did was we took fragments from the first extracellular domain and the second extracellular domain and the first intracellular domain, and we fused them together as a polypeptide. And we chose these fragments, because they contain all of the known human HER2/neu epitopes that stimulate cytotoxic T cells, plus a lot of other sequences as well. And so we made this construct, and this is the plasmid that we use that transform the cell. We've got our Listeria-lysine O PAMP fused to this chimeric human HER2/neu, various other little guys on this plasmid that help it work. And what we see when we transform Listeria with this plasmid is we get a protein produced that's just the right size for LLO fused to chimeric HER2/neu. First thing we do is we say, well, does this make cytotoxic T cells? And yes, it does. This is a standard CTL assay. We've just immunized regular mice, pulled out the spleen, looked to see whether the splenocytes will kill cells expressing HER2/neu, and this is the construct that we're talking about. It's called now ADXS31164 because it's licensed to this company, Vaxis Immunotherapies, who are developing it clinically, and that's their name for it. I have my own little familiar name that I use in the lab. Does it make tumors go away in the mouse? I mean, that's what it's all about, really, isn't it? Does it make the tumor go away? So we're using a mouse model that is a transgenic mouse model. So we think this is the most authentic and rigorous way to test cancer vaccines. This mouse actually produces autochthonous tumors between 4 to 6 months of life, and it has the rat HER2/neu gene under the control of the mouse mammary tumor virus promoter. Does this vaccine, if we vaccinate these mice, can we stop the tumors appearing? And we can. We vaccinate them every 3 weeks, starting at 6 weeks of age, total 6 vaccinations. I can't count. And then we look to see whether tumors appear over the space of a year. And this is what we see. Here is our vaccine. You can see very significant delay, well over a year. All of the non-vaccinated mice are dead. This is actually an earlier version of this vaccine, which is not quite as good. We made some improvements. And this is actually a control vector that doesn't, carrying an antigen that is not expressed by the tumor. You see they're all dead by 35 weeks, whereas these guys, we've got about 40% of them still alive well over a year. These are expensive to do. I have to say these experiments because you've got to use a lot of mice and you have to keep them for a very long time. We looked to see whether Tregs were reduced in these tumors in the mice, and they are, so we were pleased to see that. We did a number of things. Then the other thing we looked at is we all know that the primary tumor isn't what kills you. It's metastatic disease that kills you, right? And so we did this. It was a somewhat artificial experiment, but what we did was we took a breast cancer cell line that had been transducted with luciferase, and we put it into the brain. We just, breast cancer mets to the brain. And there's always an issue about with cancer immunotherapy. Can T cells cross the brain and eliminate metastatic tumors in the brain? And the answer is they can. So these are the mice that all developed tumors. As you see, quite large tumors. You can see them glowing in the brain. And these are the guys So these got the control vaccine, and these are the guys who got the HER2/neu vaccine. And you can actually quantitate this luminescence, which we've done here. And you can see here's the control, and here's the control, and here's the vaccine. So we've managed to control tumor growth in the brain, which we're pretty excited about. So we are taking this into the clinic for a wide number of tumors. There's a phase 1 clinical trial about to begin, and it's recruiting patients with any HER2/neu expressing solid tumor, including breast cancer. But we don't have any data on that. Avaxis has a pipeline, a very robust pipeline of Listeria-based vaccines, both to HER2/neu expressing tumors, but also to prostate. And this is a whole bunch of them, which are all using Listeria that targets HPV 16E7 for various forms of various stages of cervical cancer, head and neck cancer, anal cancer. So I'm going to pass this over to my colleague, Nicola Mason, at this point. She is going to tell you about her studies using this vaccine, which was developed for human breast cancer, in osteosarcoma in the dog. So why dogs? Well, first of all, they're not mice. That's a really good reason to use them. They are large, outbred omnivores, just like us, right? They have a similar digestive system. This is kind of relevant if you wanted to human-like if you want to use Listeria as an oral vaccine, which we're not currently. The human HER2/neu is 92% homologous to canine HER2/neu. It's 87% homologous to rodent HER2/neu, but we know that the human HER2/neu is effective against HER2/neu expressing tumors in mice, so we reason it would be for canine HER2/neu. 40 to 60% of canine and human osteosarcoma express HER2/neu. And a really important reason to do is people love their dogs, and they're prepared to. I can see Nikki cringing over there, but she's like, they love them. It's amazing. I mean, Nikki has people who bring their dogs in their Learjets to Penn to get treated. I'm not making this up. You can't make this stuff up. Okay, so that's me. Acknowledgements. University of Pennsylvania, Perelman School of Medicine. The people in my lab who worked over many, many years on this HER2/neu vaccine. Avaxis Immunotherapies, I have to tell you, I have a significant financial interest in this company, of which I am the scientific founder, therefore you cannot believe a word I say. But Avaxis Immunotherapies is the company that is bringing this vector into clinical trials. Phase 1 trials are HER2/neu, but we're in phase 3 trials for the HPV vaccine, so thank you. I'm not going to take questions. So we'll hopefully have some time after Nikki's talk for questions about this technology.